Your final gel image should look like this. Lanes 2 to 5 contain our DNA samples that have been successfully amplified and have undergone restriction digest, which will help us to distinguish between pathogenic and non-pathogenic DNA sequences. The piece of DNA we amplified from our samples is 400 base pairs long. When cut by the restriction enzyme, it should be cut into two pieces, one that is 250 base pairs long and one that is 150 base pairs long. You can see that in our negative control, the harmless E. coli DNA fragment is 400 bases long. But in the positive control, the pathogenic E. coli DNA has been cut into two smaller pieces. As you can see, our lettuce DNA, sample 3, has a 400 base pair band and matches the harmless E. coli strain. But the tomato sample, sample 4, has two shorter bands and matches the pathogenic E. coli sample. This means the tomatoes used as ingredients on the food trucks are the food poisoning culprit. If your gel does not look like the sample image displayed here, you can troubleshoot what went wrong by going over the most likely potential pitfalls. If you see only 400 base pair bands in your gel and no 250 or 150 base pair bands, it is likely that your restriction digest failed. This may be due to the restriction enzyme being left at room temperature for too long or not being added to the reaction. If the digestion is inefficient and only some of the PCR amplicons are cut, you may see 3 bands in the pathogenic lanes of 400, 250, and 150 base pairs in length, corresponding to uncut and cut pieces of DNA. This is okay, and you can still interpret the results. The presence of the 250 and 150 base pair bands allow you to definitively say that pathogenic bacteria were present. It is expected that you will see fuzzy bands at the very bottom of your gel lanes. These are primer dimers, a result of the short PCR primers binding to each other rather than the target DNA. If you see no bands in the sample lanes, but do see the DNA ladder, it is possible that your PCR amplification failed. Make sure that all components of the PCR reaction are added and that the correct number of cycles is performed on the miniPCR device. Seeing no bands in the gel, or in 1 or 2 lanes of the gel, could also be an indication that gel loading failed. If samples are not added directly into the wells, or the pipette tip pierces through the bottom of the gel, they may float away and produce no bands on the gel. If you see no bands and no ladder, it is possible that your gels were exposed to light during storage.